Hello everyone, and welcome to my virtual poster presentation. My name is James Lee, and I'm currently a master's student at Laurentian University. Today, I'm going to be giving a brief introduction on my current project titled Catecholamine Biosynthesis and Regulation in PC12 Cells, the Influence of Androgen and SRY. Before I begin, I would like to disclose that I have no affiliations with any organizations, and that this research received financial support from Nossum University and from NHRC. This project focuses on catecholamines. Now, what are those? Well, catecholamines are a class of neurotransmitters and hormones, and these include noradrenaline and adrenaline. Catecholamines play a role in influencing blood pressure and are a key component of the stress response. Increased levels of catecholamines have been linked to the development of hypertension, also known as high blood pressure. And hypertension prevalence and sympathetic nerve activity are typically higher in men under the age of 50 compared to age-matched women. However, this trend is reversed after the age of 60 in postmenopausal women. Additionally, men have higher systolic blood pressure changes and urinary epinephrine changes during stress compared to women. These sex differences in hypertension rates and stress response are thought to be attributed in part to sex hormones as well as other factors, such as genes on the Y chromosome. Two factors of note are androgens, which are the primary male sex hormone, with testosterone being the most notable androgen, and SRY, which is the gene that codes for the sex-determining region Y protein and is found on the Y chromosome. Both androgen and SRY play a role in the development of male characteristics, and they are thought to be linked to blood pressure regulation and hypertension. Currently, the mechanism by which androgen and SRY regulate catecholamine biosynthesis is poorly understood, and so this study utilized PC12 cells to investigate the effects of androgen and SRY on catecholamine biosynthesis. With this in mind, we had two main objectives for this project. First, we wanted to establish whether PC12 cells are a viable model for androgen studies. PC12 cells are rat adrenal cells and are a commonly used cell line model for catecholamine studies. Classically, androgens act by binding to androgen receptors, which then move to the nucleus to influence gene expression. But previous literature is conflicted about the presence and functionality of androgen receptors in PC12 cells. For our second objective, we wanted to look at the effects of androgens and SRY on the gene expression of enzymes responsible for synthesizing catecholamines. And so, in this study, we tested PC12 cell cDNA and rat adrenal tissue cDNA to see if androgen receptor and SRY mRNA transcripts were present in PC12 cells. RNA was extracted from the cells and converted to cDNA, and we used RT-qPCR to amplify mRNA transcripts for androgen receptors and for SRY. The final products were then run on a TAA gel for visualization. We then treated PC12 cells with various concentrations of R1881 and DHT, which are just different types of androgens. We extracted RNA, converted it to cDNA, and then used RT-qPCR once again to measure the gene expression of the enzymes that help synthesize catecholamines. These enzymes of interest are tyrosine hydroxylase, or TH, which converts L-tyrosine to DOPA, dopamine beta hydroxylase, or DBH, which converts dopamine to noradrenaline, and phenylethynolamine and methyltransferase, also known as PNMT, which converts noradrenaline to adrenaline. Now, this study is still in its early stages, but our preliminary results show that androgen receptor mRNA and SRY mRNA are present in both PC12 cells and rat adrenal tissue. However, androgen receptor mRNA expression was significantly lower in PC12 cells compared to adrenal tissue. We then continued with treating PC12 cells with the androgens R1881 or DHT at various concentrations. And in our experiments, neither androgen treatment had an effect on TH, DBH, or PNMT mRNA expression. Although PC12 cells express androgen receptor mRNA, the low expression of androgen receptors may have caused a diminished response to androgens, thereby reducing any potential androgen receptor-mediated changes in gene expression. To investigate this further, our next steps involve utilizing CRISPR activation technology to upregulate androgen receptor and SRY expression in PC12 cells. CRISPR activation will be done using lentiviral-mediated delivery of a dCas9 VP64 plasmid and the corresponding guide RNA for androgen receptor and SRY. We will then repeat the experiments that we have done, and in addition, we also plan to run catecholamine ELISAs to measure the level of dopamine, noradrenaline, and adrenaline released in the cell culture media. Understanding the factors that contribute to hypertension could lead to improved management and prevention of the condition, and the experiments in this study focus on the interplay between catecholamines, androgen, and SRY. The results from this research will provide insight into the influence of sex hormones on catecholamine biosynthesis and the stress response, which may help to explain the sex differences in hypertension prevalence. Thank you very much for listening.